Welcome to my project bazaar.com. Let me discuss about the project Optical-Driven Non-Local SAR Despeckling. Let me discuss about the abstract of the project. Image denoising is an important image processing task, both as a process itself and as a component in other processes. Very many ways to denoise an image or a set of data exist. The main properties of a good image denoising model is that it will remove noise while preserving edges. Traditionally, linear models have been used. One common approach is to use a Gaussian filter, or equivalently solving the heat equation with the noisy image as input data. That is a linear second order PDE model. For some purposes, this kind of denoising is adequate, while some denoising approaches such as bilateral filter, log and NLM estimate each pixel separately, fusing other similar neighborhood pixels. Some other recent state-of-the-art patch-based methods such as BM3D and plow denoise a group of similar patches together. These patch-based methods are inherently limited in performance. In this paper, optical image is added with SAR image to make the speckled SAR images. Both clustering and denoising are performed on image patches. Then the despeckling process implemented based on PCA with Weiner filtering. First, perform the clustering process to resist the influence of noise by reducing the dimensionality. Principal component analysis is implemented to reduce dimensionality, and it leads to perform the k-means clustering algorithm in order to predict the noise ranges effectively by applying genetic algorithm. For denoising process, Weiner filter denoising model proposed. Finally, obtain the denoised medical images or the SAR images. Let me discuss about the flow diagram for our process. Here, input image are taken from the dataset folder. Here we take the SAR image as the input image. In the noise image stage, an image is corrupted by optical image. One goal is in image restoration is to remove the optical image from the SAR image in such a way that the original image is discernible. Optical image is the apparent reproduction of the object formed by a lens or mirror system from reflected or diffracted light waves. Next, in the dimensional reduction stage, we used log transformation and PCA domain. Some variables are not normally distributed and therefore do not meet the assumption of parametric statistical test. Many variables in biology have log normal distribution, meaning that after log transformation, values are normally distributed. If the data shows outlier at the higher end, a logarithmic transformation can sometimes help. The logarithm function tends to squeeze together the larger values in your data set and stretches out the smaller values. Next, in the PCA domain, principal component analysis. PCA is the simplest of the true eigenvector based multivariate analysis. Often its operation can be thought as a revealing the internal structure of the data in a way that the best explains the variance in the data. If a multivariate data set is visualized as a set of coordinates in a high dimensionality data space, PCA can supply the user with a low dimensional pictures, a projection or a shadow of this object when viewed from its most informative viewpoint. This is closely related to the factor analysis. Factor analysis typically incorporates more domain specific assumptions about the underlying structures and solves eigenvectors of a slightly different matrix. Next, in the k-means clustering algorithm. K-means clustering is a method of vector quantization originally from signal processing that is popular for cluster analysis and data mining. K-means clustering aims to partition an observation into k cluster, in which each observation belongs to the cluster with the nearest mean, serving a prototype of the cluster. This results in a partitioning the data space into Varney cells. The problem of computational difficult, however, there are efficient heuristic algorithms that are commonly employed and converges quickly to a local optimum. These are usually similar to the expectation maximization algorithm and the mixtures of Gaussian distribution via an informative refinement approach employed by both algorithms. Additionally, they both use the cluster centers to model the data into a iterative refinement approaches. However, k-means clustering tends to find clusters of comparable spatial extent, while the expectation maximization mechanism allows clusters to have a different space shapes. Next, in the optimization stage, here genetic algorithm are proposed. Algorithm is stated with a set of solutions called population. Solutions from one population are taken and used to form a new population. This is motivated by a hope that the new population will be better than the old one. Solutions which are selected to form a new solution are selected according to their fitness. The more suitable they are, the more chances they have to reproduce. Generate random population of n chromosomes, evaluate the fitness of each chromosome, and create a new population by repeating the following steps until the new population is complete. Select a new parent chromosomes for a population according to their fitness. Next, with a crossover probability, crossover the parents to form a new offsprings. With a mutation probability, mutate new offsprings at each locus. Place new offsprings in a new population. Use a new generated population for a further run algorithm. Next, in the finally filtered algorithm is performed based on the Wiener filter. The Wiener filter is a filter used to produce and estimate the desired or target random process by linear time invariant filtering of an observed noisy process, assuming known stationary signal and noise spectra and additive Gaussian noise. The Wiener filter minimizes the mean square error between the estimated random process and the desired process. The goal of the Wiener filter is to compute the statistical estimate of an unknown signal using a related signal as an input and filtering that known signal to produce the estimate as an output. For example, the known signal might consist of an unknown signal of interest that has been corrupted by additive noise. The Wiener filter can use to filter out the noise from the corrupted signal to provide an estimate of the underlying signal of interest. Finally, the result will be obtained, which is the removing of the optical image from the SAR image. Let me discuss about the running procedure for our process. Select the main.mpl in the current folder, right click that file and run for executing this process. This is the main window of the denoise SAR image using principal component analysis based Wiener filter denoising model. By pressing the input image button, we can browse the SAR image from the dataset folder. You can select any one images from the dataset folder. Here I display the SAR image in the axis 1 plot. By pressing the noise image button, we can browse the optical image from the dataset folder. Select any one optical image in the dataset folder. Here I display the optical image. Here I display the combination of input image and noise image, that is SAR image and also optical image. So here we got the noise image. By pressing the sub images button, we can segment our noise image into many regions. Here I display the segmented images. And by pressing the log transformation button, we can perform the logarithmic of our noise images, so that here the dimensions of the noise image will be reduced. Here I display the log transformation image. By pressing the PCA domain, we can extract the PCA features for our noise image. By pressing the feature extraction, we can extract the feature with the help of PCA features. This is the feature extraction in a plot format. Here I display the feature values. By pressing the k-means algorithm button, we can perform the clustering operations of our feature values. Here I display the clustered group. By pressing the denoise patch, we can perform the genetic algorithm operation. Here the best value and average value are displayed. The average values are goes and reached to the best value. This process will be repeated for entire subregions of our images, so that we can obtain the best result of our noise image, the removal of image, so that we can obtain the SAR image, that is removal of noise images. Finally, we obtain the noise result for one patches, that is one segmented regions, and here I display the combined combination of denoised patches for entire segmented region, and here the PSNR value and values are for initial and final value displayed in dB, and here the PSNR response for existing and proposed system. Here the iteration level, and here the PSNR in dB are performed. Finally, the SAR image are denoised effectively with the PCA domain based Wiener filter approach. Thank you.